Okay, if you go into Canvas, I put in some more penultimate lectures, catching up, because we didn't get through everything last week that I thought we should. So I have one section here where it's from chapter 13. This is looking at linkage between genes. The idea that if you have genes that are on the same chromosome, then they're not inherited independently of each other. If you imagine you've got a chromosome that's like this, and there's two genes that are right next to each other, whatever versions of alleles that are on these, they will tend to be passed on together into the next generation, because they're only occasionally going to be split up by a crossover event. The closer together they are, the less likely they are to be split up. And so generation after generation, you'll probably get two versions of genes that are very close together, continuing to be passed on together, and then occasionally broken up by crossing over. That sort of thing was studied in fruit flies, and actually we were able to use the results of those fruit fly studies to start to map fruit fly chromosomes, and that began 100 years ago, way before we had DNA technologies. That was even before we knew what the structure of DNA was, even before we knew that DNA was the carrier of genes. We were actually mapping out the locations of genes along chromosomes, which I think is pretty amazing to think about doing that in 1920. But that's what happened, and it was based on this linkage between different genes, producing generation after generation of fruit flies, and seeing how many of them had the parental types of chromosomes and how many had recombinant ones that had been mixed up by crossing over. That allowed us to tell how close together the genes were. So you can see what I have on that in this particular mini lecture. It's 11 minutes. I apologize for the poor sound quality. I need to get a better microphone. And then I have another one here on DNA replication. This is doing part of your chapter 14 stuff. So I hope that before you come in to do the test on Thursday, everybody has a chance to view these, or else you especially study these materials hard. Since we didn't have enough time last week to get to them, I decided to do it that way. So you can watch these video lectures, and hopefully it's almost as good as being here, hopefully. So I have those in there. And otherwise, I think what I want to do here is show you the things from chapter 15. That is what DNA is for. We got up through chapters 11 and 12, where 13 we're looking at how DNA is inherited, how genes are inherited. 14, we're looking at structure of DNA and how it's replicated between the generations. 15 is where we're finally seeing what DNA itself does. And that's what we've been kind of dancing around. Now we get down to the real meat of this business. So let's see what it is that DNA does, how we actually read the directions in it to make a protein. So how do we get from a DNA to a molecule to a protein? Well, we have these genes that are located within our different DNA molecules, within our different chromosomes, usually a few thousand or so bases or nucleotides long. I tend to use the words bases and nucleotides interchangeably. They almost mean the same thing. And what will happen is you'll have a sequence of these nucleotides or bases that's coding for a polypeptide. Polypeptide then is the beginning of making a protein where you join amino acids together. It takes at least one polypeptide to make a protein, sometimes several of them together, like we see with hemoglobin. But most of your DNA is not actually coding directly for proteins. It's only around 1%. We have an enormous amount of non-coding DNA. A lot of that is involved in regulating the genes. That's not all of it, but that's at least what part of the non-coding DNA does is that it influences gene expression. It will control when certain genes are able to be read, which means that you're actually making proteins from them. And then if you're looking at a chromosome, on average, they'll have about 1,000 genes. Big chromosomes will have more. That stands to reason. But what we have 23 unique chromosomes, basically, and we have about 20,000 genes. So it's roughly 1,000 per chromosome. A big chromosome like number 1, of course, has more. Little one like the Y chromosome just has a handful. Now, this becomes what's been called the central dogma for molecular biology. This idea that genes are made out of DNA. They lead to the production of proteins, but they do so by producing an intermediate molecule. A gene that's in DNA leads to production of an RNA molecule, specifically what we call messenger RNA, and that is what gets read by a ribosome to build a protein. So these ribosomes that we have in our cells, they're actually located out in the cytoplasm of the cell, outside the nucleus. The DNA, where's that located? Inside the nucleus or in the cytoplasm? It's in the nucleus. So somehow we have DNA in the nucleus, proteins being made out of the cytoplasm. What you have is this RNA that carries a message out of the nucleus to tell the ribosome what to do. Messenger RNA, that's what the M stands for. So it's carrying a message from the nucleus. And so what we have are two major steps. One of them, transcription. With that, you have to create this mRNA molecule. You're creating that inside the nucleus as a gene is being copied. And it leaves the nucleus, tells the ribosomes what to do, and they build the polypeptide. Translation. So transcription followed by translation. They are happening in two different places in our cells. Now, if it's in a bacterium, they don't have this separate compartment with the nucleus. So these are happening basically right next to each other in the bacteria. But in our case, they happen in two different locations at different times. And we'll mostly be looking at eukaryotes, namely us. So mRNA, again, is the messenger that is carrying this set of instructions out to the cytoplasm, out to the ribosomes. There are many other functions that RNA has. And for the different functions, we put different prefixes on RNA. This is just the first one you'll see. Now, regarding the RNA, it's very similar to DNA, almost the same. One slight difference is that it's made out, it has these 4 bases like DNA does, and 3 of them are the same. Adenine, cytosine, and guanine, the bases are identical, but there is a difference where RNA doesn't use the thymine base. It uses a different one we call uracil. It's almost identical, but it is slightly different. So RNA is using A's, U's, C's, and G's. DNA, it's A's, T's, C's, and G's. Now that T and U difference can help you if you see a sequence of letters and you got a lot of the others, but then you see a bunch of U's in it, you should realize that that's an RNA sequence. If you see T's, you know it has some DNA in it. If you're seeing T's and U's, then it's probably some of both. So that can be a clue if you're trying to figure out something from a question and you get a little oriented. Look for the T and U difference, and that can give you information. Now, RNA also has a slightly different sugar in its nucleotides. Remember, each nucleotide has a phosphate, a sugar, and a base, which has a lot of nitrogen in it. These are the 4 bases. The phosphates are the same, but then the sugars are different. RNA uses a ribose sugar, and that has another oxygen atom. It has a hydroxyl group, and that makes it more reactive. So the RNA has this more reactive sugar involved. DNA doesn't have as many oxygens, so it's deoxygenated, deoxyribose. So you take a ribose sugar and take that oxygen away, you're getting rid of the hydroxyl group, and you end up with a less reactive sugar, on the DNA. So DNA is more stable, which is great if you're using it for long-term storage of information. But RNA actually does stuff. RNA actually is central in building the proteins, so it is a much more reactive molecule. The ribose sugar with the reactive hydroxyl helps with that. So these are some differences, subtle but important differences. One difference in the bases and one difference in the types of sugars that these nucleotides have. But as I was indicating there, RNA does a lot of stuff. DNA basically just sits there. It's a big repository of information. But you need to get proteins made, and that takes a lot of different molecules that are actually coordinating and doing activities. And a lot of those are RNA molecules. So we tend to think of the active molecules in us all being proteins with enzymes. Well, mostly that's true. But in this case, when you're talking about protein synthesis, RNA molecules are also very active. So we get the messenger RNA and a lot of other roles that RNA takes on. We'll be seeing those. Here's an RNA nucleotide. So this would be the U nucleotide. It has the phosphate. Notice again, negative charge, which is how the electrophoresis can separate out the DNA by grabbing on to those negative charges. Then here is the ribose sugar. DNA would not have that oxygen. It would just go carbon to hydrogen. This is a more reactive part of the RNA nucleotide. And then that's the uracil base. By contrast, here's the thymine base out of DNA. You can see it's almost the same, but this one goes carbon-hydrogen, this one has an extra carbon. The ring is otherwise the same. So you've got a slightly different looking base there taking the place of thymine. So as to where things are going on, you have the genes located on your chromosomes, which are inside the nucleus, if we're talking about us, eukaryotes. But the proteins are being synthesized in the cytoplasm, so we have to get the information for making a protein or polypeptide from one place to the other. And that's what the RNA does, messenger RNA. It takes a copy of a gene, which leaves the nucleus, and then guides ribosomes to start making those polypeptides. So if you have this arrangement, you've got a nucleus, it's got your chromosomes, has all these DNA molecules. What you're doing is identifying a section of the DNA molecule that has a particular gene on it, and that section is getting copied into a single strand of messenger RNA. Messenger RNA is single-stranded, so it's only copying one of these strands of the DNA. It doesn't need to copy the second one because really each strand here has all the information necessary due to the complementary base pairing. You have A's with T's, C's with G's. You only need one of those strands and you have all the information. So we make this single copy of the messenger RNA, single strand. It leaves the nucleus, and then ribosomes attach to this, and they start using the information there to make a protein out of the amino acids. So if you're looking at a prokaryote, well this is all happening within the cytoplasm without any sort of spatial separation of transcription and translation. That means that you could actually, if you're a bacterium, you can actually be busy making the messenger RNA, and even before it's done, a ribosome attaches and starts making the protein. So this can be spooling off the DNA molecule, and already a ribosome is attached and is busy building the protein. That can happen if you're a bacterium. Bacteria do things fast. If you think about cell division, how long does it take an E. coli cell, a bacterium, to divide and produce two new ones? It's quick, about 20 minutes. That's an example of just how rapidly bacteria can operate. So they can double their numbers and double again and keep doing that and go from one cell to billions in a few hours. It's partly because of the extreme quickness that they're able to bring to these kinds of steps. In our case, we have a little more of a slow and steady approach. You have the nucleus. It has the DNA molecule. You copy the gene, and that's copying an entire section of the chromosome from what we call the promoter all the way to a termination region. So you're copying the gene plus some stuff on either side of it. And you get this pre-RNA, and it actually gets processed, generally gets shorter, and we'll see later why that is. But we go to eventually having this finished mRNA, fully processed mRNA, leaves the nucleus, and then a ribosome goes to work. And the ribosomes could be coming on here many at once, following each other. So one can start at the beginning, work its way down the gene, be building a polypeptide, and once it's clear of the start, another could attach and start doing the same thing. So it's possible for you to be making many copies of the protein at once. But those are some important differences between prokaryotes and eukaryotes right there. But here's what you get. If you've got the DNA molecule, double helix, well, one side serves as a template for building the messenger RNA. What happens is this double helix unzips. You have enzymes that actually break the hydrogen bonds and spread this out. And one of the strands simply gets out of the way. The other strand serves as a template. That would be the darker blue one here. Notice there we've got a T, and it pairs up with an A with this RNA sequence. Imagine this is the messenger RNA. Then T pairs with A pairs with U. U is taking the place of the T base in DNA. So once we get to RNA, we're talking about U's in place of T's. U is paired with the A, C pairs with G, just like normal, T pairs with A, and so on, but then here we've got several A's in a row paired with U's. So you get this messenger RNA that's copied. It leaves the nucleus, and it provides enough information in this case to make the world's shortest protein, just 3 amino acids. That is about the world's shortest protein, for real. So you have one of these 3-letter groups here serves as a way to say, start building this sucker and begin it with a methionine. That's one of the amino acids. You have 2 more amino acids right there, lysine and phenylalanine. How many of them are there total in us? How many amino acids do we use? 20. So that's the typical number. And so there we've got 3 of them showing up. And what we have is that 3 of these letters in a gene, starting up there with the gene, now in the messenger RNA transcript, 3 of the letters provide enough information to say which amino acid goes in, and then 3 more tell you what the next one is, 3 more tell you the next one, and then you have to tell the ribosome that it's done making the protein. And so you have finally the stop codon, and this tells it to stop work. So what you have are 3 amino acids joined together, and then it's ejected from the ribosome. That's about the simplest possible thing you could look at for this, because proteins don't get any smaller than this. But again, this process is happening in the nucleus. This is happening in the cytoplasm connected to a ribosome. Questions for me here so far? Questions on this? And we'll have a sip of coffee while you think about it. Okay. Well, we have different languages that are involved here. One is this nucleic acid language, which has 2 slightly different versions. you could think of these being the difference between American English and British English. You know, they're practically the same, but there are a few differences that happen. In this case, the DNA and the RNA languages, they're both nucleic acid languages and nearly identical. They have 4 letters, 4 different versions of each of these bases. So going from one of these to the other is a fairly small process. Transcription, you're still staying within the nucleic acid language. Translation is harder. Just think about the difference between you writing down words, and I'm speaking in English, you're writing down in English, and that's not a very complicated transition to go from one to the other. But if you're actually having to translate it into a different language, then that's a more fundamental shift. And that's what happens when we go to the protein language, the amino acid language, because that is taking us to a language using 20 different amino acids. And so it's a fundamental change going from 4 nucleotides to 20 different amino acids. And that is the translation. It's harder, it's more complicated, as we will see. Well, one thing that happens here, you have to consider how do you end up actually specifying 20 different amino acids when you only have 4 different versions of the nucleic acids to use to identify those. Short film here will show how that was worked out, the math of it. And let's hope we've got enough audio here.
Once the DNA double helix had been discovered, the next big challenge was to work out how the 4 letters of DNA could code for each of the 20 amino acids that make protein. The first question was, how many DNA letters coded for each amino acid? If it was 1 DNA letter for 1 amino acid, then you could only code for a maximum of 4 amino acids. 2 letters in every possible combination could code for up to 16 amino acids. Still not enough. But 3 DNA letters provide more than enough combinations to code for all 20 amino acids. So 3 was the answer. It was a triplet code.
So with that in mind, if you're just using 1 letter of a nucleic acid, you can only specify or choose between 4 different amino acids. Or you could get 16 if you're using 2 of them, because there's 16 different 2-way combinations. How many different 3-way combinations are there? How many different 3-letter codes could we come up with? More than that, more than 20.
64.
64. 4 to the first power is just 4. 4 to the second power is 16. 4 to the third, there's 64 different 3-letter combinations. And yet, now that gets interesting because there's only 20 amino acids. So really, you would only need 20 of these different combinations, plus 1 telling you to stop, so maybe 21. And yet, we've got 64. So what happens with all those extras? We'll see. So we've got that triplet code. That's how we're actually specifying the amino acids using these triplets. You have DNA triplets, which are these different 3-letter sequences within a gene. They are then transcribed to make a codon. So if you have something like an AAA in the DNA, it'll get turned into an AAA codon. If you had, say, an AAT in the DNA, what would the codon be? if you were starting with the DNA reading AAT, what's the messenger RNA turn into? AAU. That would be the codon. And so you would have, say, the codon AAU, which is telling the ribosome, okay, here's your directions for putting in an amino acid. Well, this whole big scary table is what was worked out through decades of Nobel Prize winning work following the discovery of the structure of DNA going back to Watson and Crick. So following that, you had decades more work to figure out really how are we getting from this messenger RNA into proteins. And we end up with this big scary table which comes in different forms. This is the one I'm giving you. What we have in here are the 64 different messenger RNA codons. That's what these are. Notice there's no T's in these, just U's. These three-letter groups that are capitalized, that gives all 64 of the different codons that can be on the messenger RNA. This would be the information that's getting to the ribosome. And what we have here are the amino acids that the ribosomes will put into a growing chain of amino acids based upon the information that came to the ribosome. So if the ribosome is looking at the messenger RNA and there's a CAA codon, it knows to put in a glycine, nothing else, or it's not glycine, I forget which one that is, but this particular amino acid. Let's look at this one, histidine. I know what that one is. Here for the moment I'm forgetting what the full name of that one is. But this is indicating for us what happens with the translation, going from codons to amino acids. Now, I don't expect you to memorize all this junk. This is something where I'll give you this table, this same table, on one or more tests in the future. That won't be on the test that we're doing on Thursday. I guess it would be on the final exam when you see this from me on a test. And I'll give it to you. So what I'll do is I'll ask you to do something about where you have to use this, but I'm not going to expect you to memorize it. No more than I expect you to memorize all the steps of respiration. You do need to, I think, recognize that these lowercase three-letter groups are referring to the different amino acids. If you look this over, you're going to find all 20 of the amino acids showing up with these three-letter codings for them. Leucine, phenylalanine, serine, tyrosine, cysteine, tryptophan, and so on. That's what these three-letter lowercase ones are. Well, notice that one codon serves as both methionine and starch. What that means is that when you start building a polypeptide, right at the beginning of it, this serves as the beginning of translation, of building the polypeptide. And also, every time there's an AUG codon, the ribosome puts a methionine in, which means the very first amino acid in a polypeptide is always methionine. This is basically serving two purposes. The ribosome starts building a polypeptide and it begins with methionine, marking the start. And then after that, there may be more methionines, and it'll put more of those in. Eventually, the ribosome will get to the end of the transcript, and there will be one of these three stop codons. Any of these three will be sitting at the end of it and marking at least the end of translation. Any of these will work to make the ribosome stop, disassemble, release the protein, and say that it's done with its job of building a protein. This particular table, again, is something that will be on future tests, not on Thursday, but the final exam. And I'm giving you practice with it later this morning. You'll get to practice some with the same table with an activity. So I know it looks scary, but you know, it's not as hard as it seems. So you get transcription occurring inside the nucleus, where one big enzyme will attach to the DNA molecule at the beginning of a gene, RNA polymerase. This makes a polymer of RNA nucleotides. So that's the one that actually builds the messenger RNA. That's a copy of a gene. RNA polymerase will attach near the start of the gene and the DNA molecule. It will unzip the DNA, splitting this blue DNA into separate strands, creating this small transcription bubble where the DNA has been opened up, unzipped, and then that gives space for the RNA polymerase to start putting in RNA nucleotides that match up with these. If you've got a T on the DNA, RNA A fits with that. If there's an A on the DNA, it has to be an RNA U pairing with that. And C's and G's pair up like normal. So you end up building then this messenger RNA base by base as the RNA polymerase slides along the DNA, temporarily unzipping the DNA, breaking the hydrogen bonds between the DNA bases, spreads them apart, and then they close right back up. Here the RNA polymerase is moving to the right, and these have already closed back up. So the DNA can go from a double helix to separate strands and then right back. Just like zipping your clothes up and down. You get a zipper as something where we can easily disassemble clothes and then put them back together. Here, it's the same idea. And molecular biologists refer to the DNA unzipping and zipping all the time. And it's a perfect analogy. So you'll have a region near the start of the gene that's called promoter, and that's marking a binding site for the RNA polymerase. It will attach, and that will then open up the DNA molecule. It'll create this transcription bubble and begin building a copy of one of the DNA strands. It's going to move down this whole gene until it gets to a terminator DNA. That's telling it to stop the transcription. So RNA polymerase moves along, steadily moving from the promoter, heading toward the terminator. And as it does that, the RNA copy is spooling out the side of it. Eventually it reaches the termination sequence. It falls off the DNA molecule. The DNA is all back to being a double helix, and it releases the RNA. So this sort of thing goes on in our cells all the time. Anytime we're having to make new proteins, we're doing this, and that's happening constantly. Now, actually, before we get to that, I'm going to show you a movie of it. This is kind of an intimidating film, but fun in some ways. It shows a lot of details that we don't need to worry about yet. We'll later come back to some of them, but there's a lot of stuff that shows up right at the beginning of this that right now we don't need to worry about. But let's watch this whole thing. Transcription.
The central dogma of molecular biology, DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
So that was showing it actually happening in real time. That's giving you an indication of how quickly these things happen with the high quality animations that are available that I'm showing you here. These are actually showing just the actual speed of the processes. So we've got that. We will see some others. By the way, in Canvas, I put in the chapter 14 section in lecture notes, I also put in several of these videos dealing with DNA replication. You should watch those too. Similar quality animation showing DNA being copied, including the leading strand and the lagging strand, which is more complicated. You should definitely look at those videos before we do the test Thursday. You have questions for me on what we just saw? Yes?
Where the free RNA molecules are coming from?
They are circulating in the cytoplasm, or in that case, inside the nucleus. So in the nucleoplasm that's inside the nucleus, there's always going to be a supply of those there, so the cell is able to go and build copies of the genes. So they would simply be floating in suspension. They're polar molecules, so they easily dissolve in this watery nucleoplasm. And that's obviously leaving out all the other molecules that are also there, but they are finding their way into this intake hole, as they called it. And if there is one that fits correctly with a nucleotide that's already there on the DNA, then it'll get plugged in there in place. The molecules are constantly shuffling around due to diffusion at that scale. On this scale where things are just atoms wide, molecules are bouncing around very quickly, and you only have to wait a split second for the right one to bounce into place. Hard for us to imagine that everything at our scale looks like it's stationary. We don't see things doing this, but on that scale they are moving that way. Other questions on this? Okay, then let's see some more. So, something that gets more complicated is this idea of exons and introns. This is something we do, bacteria don't. This is something that's more in eukaryotes. And what happens with that is that if you're looking at one of our genes, it's actually broken up into a lot of separate sections. And what you will have are coding sections called exons, which are ones that actually are information getting to the ribosome, telling the ribosome, basically providing the codons that will tell the ribosome which amino acids to use. But these exon regions are separated by spacers, introns, that are not coding. So a gene, for us, actually has a mix of coding exons and non-coding introns that are spacing these exons out. So you might have, say,